Good afternoon, everybody. The title of my talk today was originally supposed to be on some of the deep sequencing that we'd done during this big buffalo experiment. However, we've moved on to doing the antibody titers as well, so today I'll be presenting some of that. So typically, buffalo ecology, behavior, and physiology strongly mediated by seasonal cycles in buffalo, where body condition drops due to nutritional restriction, the dry season, which occurs in the Kruger National Park between June and September, and then it's regained when the rains return and new grass growth occurs. So immunity in African buffalo varies seasonally in the Kruger Park. The innate immune responses are stronger in the dry season, and trade-offs between immunity to intra and extracellular pathogens are detectable in the dry season, but not in the wet. So buffalo is the primary maintenance host of FMD, but as you've just been told, it is mild and there are no obvious signs of FMD clinically in buffalo. So consequently, we expect that strong seasonal variation driven by resource availability in co-infection patterns and immunity in free-ranging buffalo, which may mediate FMD transmission dynamics. From previous work, we know that buffalo virus recovery in field studies has occurred in different studies. There's been from individuals for 5 years reported. An experimental study that we did, we could isolate virus over 1 year. I'll show some of that data just now. And then in buffalo populations, which were 30 to 100 individuals for 24 years, which has been challenged as to maybe those were not isolated completely during that 24 years. But nevertheless, can FMDV perpetuate long-term without reintroduction from neighboring populations? However, the frequency and titer of virus recovery decreases over time, and they can clear the virus over 15 months. So the significant number of animals that fail to maintain persistent infections for prolonged periods of time as a proportion of persistently infected animals falls after reaching a peak at about 1 to 3 years of age. Serological surveys show that in the Kruger, 98% of animals, buffalo, are exposed by the age of 2 years to all 3 sets. So FMDV must maintain a high force of infection in buffalo populations continually. So previously we did work where we infected experimentally buffalo in bomas, and what we found from this study, which I would like to focus on, is how different these sets are. Often we try to clump the sets as one sort of type of set, but actually there are 3 very different serotypes, and from this study we could show initially that if you did co-infections in cell culture with different multiplicities of infection and you did different ratios of this, you could show that SAT1 outcompeted SAT2 and SAT3. Similarly, if you did different proportions of those in cell culture, SAT1 and SAT2 with MOIs of 0.5 and 2 could outcompete SAT3. So depending on the different ratios, competition was different. In buffalo infections, when we isolated virus over time, we showed that the proportion of positive samples decreased over time at a rate that differed significantly between these different serotypes. There you can see SAT1 could be isolated for up to 400 days in an isolated buffalo herd, which we infected experimentally, whereas SAT2 could only be up till about 200 days, and SAT3 between the two. So this shows that the serotypes are not equal in the way in which they work and their dynamics, and so this we wanted to further investigate. So just to bear in mind that FMD in Africa is influenced by 2 different patterns. You have the African buffalo as a reservoir, which is persistently infected, and that has its own cycle within the buffalo. And then there's infection of buffalo calves and possibly escape then to other cloven-hoofed animals with transmission events. And that causes then a livestock infection. And then within livestock, you have your own epidemic cycle happening there. So there's 2 different cycles occurring in Southern Africa. So given the prolonged annual birth pulse, the virus might be maintained as a typical childhood infection, circulating through new susceptible calves following the loss of these maternal antibodies, with the latest born calves of that year sparking the new epidemic. But preliminary models of FMD dynamics in this free-ranging African buffalo herd suggest that calf-to-calf transmission alone is unlikely to result in long-term persistence. or that the annual epidemics among susceptible calves may be initiated by persistent carriers or novel antigenic variants. So we employed, during this project, different phases of it, and I will be concentrating on just looking at all the circulating antibodies now. And to show you, so this study is really a very interesting study in the sense that never in the world have we ever tracked buffalo, the same animals, for a long period of time and sampled these buffalo regularly every 2 to 3 months to get the full picture on how the dynamics of these buffalo actually look in terms of serology and how they transmit the virus. So just to show you, this is the map of South Africa, where the red is the Kruger National Park. That star there is the Satara area, and we have a camp there that we had 64 buffalo in there. They'd been in there for many, many years prior to the study, and it was fenced off. In June 2015, we brought in 20 new animals, and we had 2 break-ins. I know it's hard to believe, but 2 break-in buffaloes. And of course, babies were born during this period of time. So the aim was, what is the seroprevalence of the FMD in this herd in terms of ages and sex over time? What are the antibody dynamics for these sats and buffalo? How variable are the maternal antibodies and their drop-off over time? And what predicts the antibody levels in the mother and the body condition and antibody levels? So we looked at the general seroprevalence of this herd using the liquid phase blocking ELISA test for the different sats. And basically you can see that if you look at the blue line, the SAT1 has a much higher seroprevalence than the other 2, with 2 main sort of spikes occurring there and towards the end there, and then the virus is quite highly maintained there with a high prevalence. The SAT2, on the other hand, if you look at from when it began, it had a single spike, and then gradually little spikes maybe, and downward towards the end with possibly a little bit more at the end. But the SAT3 always maintained a very low seroprevalence with 1 or 2 slight spikes, but always remained at very low seroprevalence compared to the others. So if we look at the different antibody responses mounted against these 3 sats, this is just to show it's a histogram, but you can decide which graph you want to look at. They're exactly the same thing. Here what we did was we looked at when an animal is positive, how high is that positivity? At what type of titers do they reach across all the animals? And what we found is that the average titer for each animal across all these time points for each sat was different. For the SAT1s, they reached an average positive of 2.23 titer. SAT2 was just below that, and SAT3 was significantly lower at 1.94. Sex made no difference, but age in this case did, where older animals tended to have a higher titer than younger animals. So as an antibody response, the SAT1 was much more robust in its titers, intermediate for the SAT2, and very comparatively weak for the SAT3. When we looked at the duration of this antibody response amongst the SATs, we found that the antibody responses to the SAT3 are much more short-lived than compared to the SAT1 and SAT2. And how we analyzed this was basically looking at the profile of each animal. And this over here indicates the amount of times that an animal had a drop or an increase in its titer that was regarded as a positive. So when the titers were maintained above 1.7, how many times an animal actually maintained that over the 3 years. So you can see that the average amount of captures in which a SAT1 was maintained was for 4 captures, whereas compared to like the SAT3 viruses, the immune responses was only very short-lived with 1 capture, and then it would drop its immunity again and rise up. So it was very short-lived. Okay. And then for the next aspect we looked at was the antibody titers to all the sats that are highly dynamic in buffalo and how they lose their protective antibodies over time. So this was analyzed. We report here the incidence of the dips in antibody titer. So this is the loss of seropositivity, and the figure shows that the number of these observed dips per animal across the 3-year period. So the SAT1 titer was relatively stable compared to SAT2 and SAT3, which the dips were well below the protective levels more often for these 2 serotypes. And the loss of this serostatus was not affected by sex or reproductive status of the buffalo. For SAT2, the younger animals and those in a worse body condition were more likely to drop their antibody titers under those conditions. Whereas with the SAT1 and SAT3, your antibodies showed a drop when there was low total immunoglobulins in the whole animal. So which shows there are different parameters to these different sets and how they are able to mount a response under different conditions. So buffaloes frequently change from negative to positive serostatus, and only a fraction of these rises in seropositivity can actually be explained by re-exposure to the viruses. Thank you. We report that the incidence of these rises in antibody titer in the 3-year study. And basically, we showed that the incidence of antibody rises did not differ amongst the different serotypes. The primary infection events of animals below the age of 2 years was analyzed, and it was shown that the incidence of primary infection was much higher for SAT1 than SAT2 and SAT3 viruses. And given this low to moderate force of infection in our herd, rises in antibody titer from negative to positive are often not attributed to disease transmission. And there were other biological processes, such as changes in nutritional status or immune status, that played a role. And again, there were differences between the SATs, where SAT2 and SAT3 in older animals were more likely to gain a positive serostatus. And for SAT1, with higher total immunoglobulins, were also more likely to raise their FMD titers. All SATs in this were not affected by poor body condition. All were likely to gain positivity through poor body condition. Again, we also looked at how calves lose their maternal antibodies, and we found that out of most of the cases, we could not detect this loss in SAT1 animal or SAT1 incidences, because it's most likely because the SAT1 is high force of infection within this herd, that we could not look at this loss of maternal antibodies because our period of 2 to 3 months of intercapture intervals was too close to actually detect that loss. Just looking at susceptibility at the herd level, susceptibility is distributed across all age groups of the buffalo and occurs throughout the year. Patterns in time and among age groups vary among the serotypes, and these patterns are likely to affect the dynamics of the herd and the endemic of the natural host populations. It's a similar figure, just showing the captures again for susceptibility. And then cross-reaction, we determined, was also not a major factor in our analyses, because it's well known that the SATs can cross-react with the ELISA, and we found that there was not much of an influence of this when you gained positivity with the testing. So in conclusion, seroprevalence shows differences between SAT1 with a higher seroprevalence, followed by SAT2, and SAT3 just remained low and decreased over time. The SAT serotype antibodies differ significantly, with buffaloes mounting very robust antibody titers for SAT1, intermediate for SAT2, and comparatively weak and short-lived for SAT3. SAT1 titers are stable compared to the other two serotypes, which dip below protective levels much more often. The loss of serostatus is affected by different conditions, which I discussed, and buffaloes frequently change their serostatus, but only a fraction of these rises are explained by re-exposure to viruses. So just to end off, we propose that SATs have evolved different strategies to coexist in buffalo, with SAT1 being highly transmissible and adapted to buffalo. How SAT2 and SAT3 differ needs to be further explained, but for SAT2 we don't find much virus circulating. However, we know that cattle are affected predominantly with the outbreaks in southern Africa, and so we propose that maybe antigenic changes occur that cause these cattle outbreaks. But in terms of transmissibility, we have found SAT2 to be very low amongst buffalo. Thank you very much for your time.